G'day team, in this video we're going to talk about electron microscopes. We're going to talk about the invention of electron microscopes, the technology that's actually involved in them, and then look at the two main types of microscopes, the transmission electron microscope and the scanning electron microscope. And then I'm going to talk briefly about the specimen preparation that's necessary for these electron microscopes to work. So the invention of the electron microscope, there's a few people involved, but the main person there is Ernst Rösker in the 30s. So he was involved in developing the first prototype as well as the first working model of the electron microscope. So he was able to work out this technology and basically get it to the point where it could be used at universities. Now, the thing that's different about electron microscope is instead of having glass lenses that focus beams of light, it actually uses electromagnets that focuses a beam of electrons or that are ejected from a high energy electron gun. The big benefit of using these high energy electrons over light is that they have a much smaller wavelength. So where light has a wavelength in the hundreds of nanometers, the electron beam has a wavelength down in the picometers, which are really small. This is important because when you're shining something through a specimen, how much that the wavelength or how much that wave is oscillating actually is going to affect your acuity or how small the things that you can differentiate from each other in your specimen. So by having this very small wavelength, we're able to see clearly things that are much smaller than with the light microscope. So we get both better acuity and much better magnification. Now there's two main electron microscopes. The transition electron microscopes works quite similarly to a compound light microscope, where the source of electrons is on one side of the specimen and is shone through the specimen, and then the receiver is on the other side, so that light is being transmitted through the specimen. Now this is a really good way to get very high magnification, two-dimensional images. If we want three-dimensional images, we can use a scanning electron microscope. And this works by shining the beam of electrons onto the surface of the specimen, and then they bounce off the surface into a detector. So rather than shining through, they're bouncing off. And the scanning part is that the source of electrons or the detector is actually moving around. So you get a three-dimensional representation of this specimen. Now we're still going to get very high resolution, but we're not going to get the same amount of resolution as we are for the transmission electron microscope. Whilst we get this really high magnification and high resolution of our electron microscopes, there are limitations, and these limitations mainly come through the procedures and techniques that need to be employed to prepare the specimens for the electron microscope. So one thing is that this electron beam, because the electrons are so small, they actually have to be shot through the specimen or off the specimen in a complete vacuum, because they are so small that if there were air particles in that area, the electrons would actually bounce off those air particles. So those air particles have to be sucked all the way out to provide a vacuum. We also have to have these specimens, because again they're so thin, dried and fixed to a medium. So this is a chemical drying process, which means that it's not going to work for some biological materials. It's actually going to destroy them. We also have special dyes. So while we have optical dyes in a light microscope, because we're looking at electrons and electrons diffracting in different ways here, we actually have to use these special dyes that work with an electron beam, which are quite hazardous and more hazardous than the ones used in a light microscope. Another thing that we've got to do is make sure that the electrons, particularly in SEM, are able to bounce off the specimen. So an electron's so small that usually it would actually penetrate something if it was shot at it. But because we want it to bounce off into our detector, we need to provide something for that to bounce off. Now, a lot of the time, this means that the specimen needs to actually be coated in gold plate so that the electrons can bounce off that gold plate. So for this reason, we can't see living processes with an electron microscope. We can see the structures, but we can't see what those structures are actually doing on that subcellular level because the specimens actually have to be dead and fixed and all this sort of stuff so that we can view them with the electron microscope. In this video, we've talked about the invention of electron microscopes by Ernst Ruska, the technology having a high energy electron beam focused by electromagnets rather than a light beam focused by lenses. And we've talked about the two types of electron microscopes, the transmission electron microscope, which gives two-dimensional images from shining the electron beam through the specimen, and the scanning electron microscope, which gives three-dimensional images from bouncing the electrons off the specimen. And then we've talked about some of the different preparation techniques that are required to view specimens with an electron microscope and how this means that those specimens, the biological specimens, can't be living and therefore we're not able to see processes, only structures. Thanks for watching. Peace out.